Welcome to our podcast. Today we invite the NTHU iGEM team to share their project with us. I'm Kevin.
Hi, I'm Rebecca, and I'm the team leader of the NTHU iGEM team.
So your project is about environment issue. How did you come up with the idea?
This is how our inspiration comes from.
If you take a walk through our campus, you can see roads lined with towering pine trees. Pine cones were scattered underground, under the shade of trees, covered with shedding pine needles. However, we noticed that some twigs of pine trees have droopy and brown needles. This symptom is unusual to evergreen plants, and it turns out to be a plant disease, the pine wilt disease.
Pine wilt disease is a serious epidemic among pine trees. It happened not only in our country, Taiwan, but also around the world, especially in Japan, China, Europe, America. The seriousness of this problem is that pine trees hold plenty of economical value. The cones, trunk, pine nuts, and rosin can serve as industrial material and ingredients. Most importantly, pine forest provides a natural environment for diverse creatures. Losing pine trees will cause many problems that we've never thought of. In Taiwan, pine wilt disease has wiped out over 5,000 hectares of pine forest. It also causes 20 million economic losses in the world.
Wow, it sounds like a serious problem.
Yes, it is. And it is harmful to most species of pine nuts. Nowadays, we don't have an efficient solution.
But how will nematode cause the pine wilt disease?
To solve the pine wilt disease, we must understand how the whole thing happened. There are three main characters involved in the spreading of this disease: the pine wilt nematode, Versiphellancus xylophilus, the host pine tree, and the important vector insect, the longhorn beetles. So, the story starts with the longhorn beetle happily looking for its favorite food, the pine tree. When the beetle finds a healthy pine tree, the nematode inside the beetle will spread onto the pine tree. The nematode will then feed on the epithelial and racing canal cells of the pine tree. Then they start to reproduce and become more and more. During the time nematodes migrate throughout the pine tree, the tree begins to show wilting symptoms. The poor injured pine tree will release some chemical compounds, which attracts the longhorn beetle to visit again and lay their egg in the tree. Then, the egg hatch into larvae. The larvae form into a pupa. Around the pupal chamber, there will be some fungi flourishes and attract nematodes to gather around the pupal chamber. While the pupa matures, the bee Xylophilus enters the beetle's body, so the beetle will now carry nematodes. Then the newborn beetle flies away to find another healthy pine tree for food, helping nematodes to spread. And of course, there must be much more than one longhorn beetle in the forest. Therefore, the pine wilt nematodes from a single pine tree will end up infecting a considerable amount of pine trees in the forest. Eventually, all the pine trees in the forest become wilt and then die from this notorious disease. Three current solutions are used to tackle it. Removing the dead trunk is labor-intensive and limited in scale. The insecticide dripping method may affect ecosystem and is unsustainable. As for the biological control, it may be uncontrollable without experts and even mistargeting. Therefore, there is no efficient solution to deal with it in reality.
So for this severe problem, how are you going to deal with it?
This year, we aim to generate a kind of pine tree that can synthesize siRNA that encodes important gene information to bee Xylophilus, such as targeting related to reproduction and pathogenicity. When nematodes uptake plant cells, they also uptake RNA produced by plant cells. Once the nematode uptake RNA fragments, an important gene of bee Xylophilus will be knocked down.
Could you please simply explain the mechanism of RNAi It sounds like it's the most important part of your project.
To illustrate the application of RNAi mechanism in our project, let's take a closer look at the transgenic plant cell. After transformation, the cell would produce double-stranded RNA of nematode genes. A protein called dicer will then cleave the double-strand RNA into pieces, forming small interference RNAs. These siRNAs are now ready for mediating the RNA interference. As we inoculate nematodes on the transgenic chylus, the nematode will suck out the cell contents as food. Meanwhile, uptake the siRNA. Inside the cell of nematode, proteins will bind to the siRNA and discard one of the double strands, forming the RNA-induced silencing complex. The remaining strand serves as a template for recognizing and binding complementary target mRNA. Once bound, the mRNA will be degraded, causing a decrease in gene expression. To reduce the pathogenicity of B. xylophilus, we choose two genes as our targets. The first one is BXPRX, which encodes peroxide redoxin. Silencing this gene will affect the reproduction and propagation of nematodes on pine trees. The second one is BXPAL1, which encodes pectidase, a cell wall digesting enzyme. This enzyme is secreted from the esophageal gland of nematodes and closely related to their consumption and migration. By silencing these two genes, the pathogenicity to pine trees of B. xylophilus would be weaker.
How about the feasibility? Any expert you consult with?
We visited Dr. Lin, who was the former PI. We asked about the feasibility of our project and how to improve it. Dr. Lin suggested that we should clarify the presentation of our result, make it more straightforward by simply comparing the modified group and the control group. Also, he offered us the idea of choosing a suitable promoter that induces the encode of our sequence in the plant, and some candidate plasmids for agrobacterium transformation. Then we asked Dr. Lan the same question, who is an expert in microbiology. Due to Teacher Lan's suggestion, we decided to construct a plasmid with the target gene separately to get a better result. We also construct a model for realizing what concentration of RNAi we should add in an attempt to kill the nematodes.
Would you mind sharing the method your team designed the sequence with us?
We will construct a plasmid that inserts sense and antisense sequence. By doing this, RNA that's transcribed from this plasmid can fold into hairpin RNA.
Wait a minute. Can you explain it more simply?
Our vector consists of three main components, which are a sense strand of the target nematode gene, NPDK intron, and an antisense strand. The original vector has only comprised promoter, terminator, which is conserved among plants. Therefore, we have to insert sense antisense strand into PBI 1-to-1 vector. Basically, sense and antisense strand are just inverted sequence. That is, they are complementary. Each base from the sense strand can match with the antisense strand. Then, we also have to insert the PDK intron. The function of the PDK intron is to promote hairpin structure formation and set up an intron loop. So, the hairpin formation starts with the RNA polymerase transcribing a linear mRNA. Because the secondary structure is much more stable than the linear mRNA, the antisense will fall back and form base pair with the sense strand. As a result, the RNA forms into a double-stranded hairpin structure, which can perform a further RNAi knockdown mechanism.
Hello, teacher. As for the plant callus cultivation, we consulted with Dr. Liu, who is a specialist in plant biotechnology. One of the biggest challenges in our project is the agrobacterium transformation, so we asked Dr. Liu about the experimental details. Dr. Liu provided us with several simplified and efficient methods for agrobacterial transformation, such as the freeze-thaw method and electroporation. Dr. Liu also suggests that the antibiotic selection followed by plant regeneration is extremely time-consuming. Therefore, focusing on the callus culture stage is much more practical to a short-term iGEM project.
How do we modify the gene of a pine tree? We use agrobacterium to methacin to transform plant cells. Agrobacterium is a plant pathogen which can infect plant cells, and the pathogenic mechanism is to transfer the tDNA region in its plasmid into the plant genome. Therefore, it's widely used in genetic engineering of plants. For the first step of agrobacterium-mediated transformation, co-culture callus and agrobacterium suspension. Here shows an agrobacterium in detail. Our engineered part is located on the tDNA region of the PBI121 plasmid. By contrast, the TI plasmid is like a toolbox. Its sequence encodes various proteins that are necessary to the tDNA transfer. When an agrobacterium is attached to a plant cell, it will transfer its tDNA region to the plant cell. The tDNA will then integrate it into the plant genome. After transformation, the callus will be selected on the antibiotics plates. As a result, the remaining callus will possess the ability to produce double-strand RNA as a weapon against nematodes.
In order to learn some basic knowledge about nematode, we consulted with professors working on C. elegans in NTHU. First, Dr. Wagner's fellow taught us how to subculture C. elegans in the lab. What's more, he also gave us some advice about our project presentation, which made our idea more understandable. Next, we visited Dr. Wang, who is also an expert in nematodes. Since C. elegans is genetically different from pinwheel nematodes, it would be better to use Basilophus xylophilus for experiment. Dr. Wang kindly introduced us to Dr. Yang from National Taiwan University, who works on plant parasitic nematodes.
Therefore, we took a little trip to Taipei for visiting Dr. Yang and saw what kind of help we can get. Fortunately, Dr. Yang does have pinwheel nematodes in her lab. First, she provided us with enough nematodes for our experiments. Besides, the student in Dr. Yang's lab also taught us how to cultivate B. xylophilus and fungus as nematodes food. Finally, with Dr. Yang's nematodes and protocol, we are ready to start our experiment.
We cultivate Basilophus xylophilus on Celanthus. First, we inoculate fungi on the solid medium as a food for the nematodes. Then we put on the nematodes and incubate at 25 Celsius degrees of culture every 7 days. Here shows how a Basilophus xylophilus look like under microscope. Because the main reason why pine tree wither is the consumption by nematodes, we calculate the consumption that would harm the pine tree first. Then we build a model to compute the harm applied to our modified pine trees by nematodes. By comparing them, we can predict the result well with a low cost. To compute the harm of nematodes, first we need to know how our modified pine tree affect them. Concentration of mRNA is related to rate of transcription and concentration of RISC. The rate of transcription is dominated by repressor protein. The repressor protein's inducer is not an RNA, thus, it should be a non-linear dependency. Unfortunately, it's impossible to determine the proper nonlinear relation between transcription rate and the concentration of an RNA because it depends on several complex mechanisms. Therefore, we use linear and highly nonlinear model to approximate it. With the toxicity model, we could build the toxicity breadth and depth model to compute the consumption. In the real situation, there is a many mechanisms involved in nematodes' life. By several discussions, we successfully simplified these complex mechanisms. We also designed several experiments to find the data that would improve our model. Therefore, we could make our model computable without losing authenticity. Here are some results of our model in different variable sets. We plot the evolution of population size and accumulation of consumption of nematodes versus time. This plot shows the result for unmodified pine trees. In contrast, this plot shows the result for modified pine trees. The red dotted line represents the consumption that would harm the pine tree. Does your team have any plan for the future experimental work?
We would like to measure the expression of nematodes gene by using the qPCR method. If after uptaking our gene-modified callus, target gene expression was downregulated, then can preliminarily prove our idea. Also, we will move on to the next page. Try our method on real pine trees. We have contacted a tree protection company, Kenred, to discuss the future experiments.
Now, let's talk about the future. In the short term, we will try to promote the productivity of siRNA in callus and improve siRNA transportation efficiency. By doing so, we could ensure that all parts provide callus the ability against nematode to some extent. In the midterm, we will strengthen the biosafety of our project. For example, design a kill switch system. Besides, we will further establish plant generation to obtain seedlings in the lab. In the long term, we hope this project could be commercialized and implemented in real-world plantations. Moreover, this method using the RNAi mechanism might be able to apply to other plant diseases.
It is a board game about synthetic biology.
Hey, look! Why our article appears at here?
Are you being excluded by others?
We sent this article to PenSci and they have published it. PenSci is now the largest scientific website in Taiwan. I think we can introduce this topic to disseminate environmental issues to the public.
Are you serious? You've never come to record our podcast? Since August 4th, we record our podcast every week, and we will post it on Facebook and Instagram. Look!
Here is the information about our podcast. In our podcast, we have a brief introduction about iGEM. We share our experience and the difficulties we encounter. If you are interested in iGEM, you will definitely love it.
Thank you for watching our podcast. See you next week at the same time.